Okay, now we will get into the pulmonary infection, because the candida can enter into a body anywhere, right? As a normal flora, yeah, endogenous or exogenous. But for the Cryptococcus or any organism from now on, yes, will be mostly from pulmonary infection, like Cryptococcus neoformans, thermodimorphic fungi, Histoplasma coccidioides. All of these listed names we found in Thailand. The green one, coccidioides immitis, this organism actually is endemic in US, in Arizona area, in the dry area. But in Thailand, we have the imported case from US. Mycoses juravexi, I think in here we will study you will be in parasite, right? But actually it is fungus. How it can go into our respiratory tract? It depends on the size. Any spore or any molecule that more than 7 just deposit in the nose. But in between 3 to 7 will be at trachea or bronchi. But less than 3 will be in alveoli or lower respiratory tract. Right? Normally the spore will not, independent in the air. It will associate with some dust, the small dust, like PM, how many? PM 2.5 will not have a fungus. PM 10. Yeah. Why I say that PM 2.5 will not have the fungus? Because most of the spore, the size will be 3 to 5, right? But PM 2.5 is very small. So the spore will attach with PM medium size or PM 10. So it can go into our inhalation. And the fungus also very fantastic. It can disguise, it can change their morphology from the small size to be bigger or to smaller, shrink and then form, and shrink and form. It depends on where they want to go. This is the wonderful factor, wonderful morphology property of the fungus. The Cryptococcus, as I told you, Phylobasidiella. This is the basidium. Yeah. And the basidiospore just stay together like a chain, like a filament. That's why we call Phylobasidiella. Okay? This is a sexual stage. And this is a sexual stage. A sexual stage, the terminology will be anamorph. Right? Sexual stage, the scientific terminology will be teleomorph. And this is the real one. Yeah, the real under microscope. Cryptococcus belong to the phylum Basidiomycota because it produces basidiospores. The blastospore of the Cryptococcus. What is the common name of the blastospore of Cryptococcus? Common name of the blastospores. Budding cell, okay? Budding cell equal to blastospores. With the high technology of the molecular, we can divide Cryptococcus into 1, 2, 3 big groups. And some scientists, they want to give the species to all of this, but we still refuse for that. So we just get only this simple way, like we call serotype A, serotype B and C, and serotype D. Okay. Now they have a serotype AB, AC, CD, whatever, let it be. Okay. We just know that most of the Cryptococcus that isolate from AIDS patient will be Cryptococcus grubii or serotype A. Somewhere or somehow you can see Cryptococcus grubii or Cryptococcus neoformans variety grubii is the same. Okay? Then the second one from its patients will be Cryptococcus neoformans variety neoformans, or we call VAR neoformans, or serotype D, D-dog. Another two serotypes are B and C. It belongs to Cryptococcus gattii, or someone called Ver gattii. Okay? For the gattii, in Thailand we have less gattii. Mostly are grubii and neoformans. I will show you how it's different between two groups. The habitat is from the bird droppings of the pottery tree. I put here the koala because now in the former time, we misunderstood that Cryptococcus will be on the bark of the eucalyptus tree, but actually not. It's just because in Australia they have a group, very active group, study of the Cryptococcus, and they have most of the eucalyptus tree, a lot. So they just understand and they isolate from the eucalyptus tree. And also koala will be eat eucalyptus leaf and live on that tree. Then the feces of the eucalyptus have the same molecular weight and molecular type of the Cryptococcus as on the tree that koala climb. So they just put in here, the habitat will be koala. But actually, Cryptococcus can be found in the bird droppings, especially pigeon, พิราบนะคะนกแก้วเขาเรียกว่าอะไรนะ parrot แล้วก็นกหงส์หยก I don't know how to call นกหงส์หยก Now not only that, now the chicken also. Then they have a opportunistic infection, human and animal. Anyone have the cat at home? Anyone have the cat at home? Cat, you are a healthy cat, right? Okay. Healthy cat is okay. But if the FIV cats, please do not feed or have the FIV cat, because the FIV cat can be the source of the Cryptococcus infection too. Okay, especially at the nose, because the cat will put the nose เขาคลอเคลียร์อยู่บนซอยที่มี Cryptococcus, then it will infect at the nose and have a very ugly lesion. And then when you hug, then you can get infection from that. You doesn't have the FIV in human, but you can get high possibility and high risk to get the Cryptococcus on FIV cat to human. Okay? Then this one can be acute, subacute, and chronic infection, as already explained before. The Cryptococcus, this is one of the wonderful property of the Cryptococcus. It can be sexual stage as a basidospore. It can be asexual stage, to be infected in our body, okay, or in another meaning, sexual reproduction and asexual reproduction spores can be the infected organ, infected propagule that we inhale to our body, or budding yeast cell with capsule and basidospore can be inhaled and cause the disease in our body. Okay, this is very special of the Cryptococcus. And how is the manifest in the patient? Pulmonary Cryptococcus inhale into the lung. Then the target organ will be brain and meninges. That is the target organ of the Cryptococcus in any species. Okay. Then after get into the central nervous system, it can disseminate. It depends on the host. It can disseminate to the brain and meninges. And not only that, it will disseminate to skin. Okay. Normally the CD4 will be very less than 100. That manifests to the skin. Osseous, where the osseous, the bone, ocular, and the scalp. So Cryptococcus is another important yeast that can cause several kinds of manifestation. For the HIV group, you see it here, in Sahara, Africa, a lot, very high. I think this is like a God ลงโทษ มั้ง Many diseases in South Africa. What else? From AIDS, then have Ebola too, right? So many diseases in South Africa. 70% will be neoformans and 30% will be Gatti. Most of the Gatti will be found in Victoria, in Canada, and in Canada. The symptom similar when you are going to have an examination: headache, fever, confused. Right? When you have a candida, right? You always like this. When you're going to have the examination, cream, right? Headache, confused. But the different thing is what? Nausea, vomit too, right? Because of the create, very stress. But the difference is that you have more weight. You gain the weight because you always eat. Yes. You gain the weight. And then sleeping. But the Cryptococcus, no, they are lost the weight. This is what is the difference. And mostly you can see reduced visual acuity related to the raised CSF pressure. So what is the specimen for the cryptococcosis? What is the specimen? What should be the good specimen? What? CSF. Oh, how to, very invasive. But the first step. Because normally when we get a specimen from the patient, we will get less invasive first, right? We will consider for the less invasive. So what is that? What is the root? Root of infection is pulmonary, respiratory tract, right? So what is the first step to get? oral cavity, oropharyngeal swab. Right? And Cryptococcus has a capsule, so normally it will attach. So that is the good part. Then to where? To pulmonary, to the lung. It will be like a ball. We call cryptococcoma. For the healthy patient, Cryptococcus will become as a cryptococcoma like a ball and just stay still. If the innate good macrophage will be killed or phagocytosis will be phagocytosed and kill the Cryptococcus. But for the low innate immunity, the Cryptococcus will get into the macrophage. Then, what else? Think. When the Cryptococcus, we inhale a lot of Cryptococcus in our body. After we inhale innate immunity, for example, macrophage, will phagocytose and just phagocytose. They want to kill them, but with a high amount of Cryptococcus, it cannot kill. Cryptococcus cannot be killed. So, macrophage will become like a truck, like a horse, big horse, like a Trojan, like a big vehicle เหมือนกับคอนเทนเนอร์ใหญ่ๆ Then Cryptococcus inside will be moved together with the macrophage to where? Macrophage will go to where? From the lung then to several places in the body, right? Into the bloodstream. Yeah? In the bloodstream, Cryptococcus itself also can become smaller, then try to get into between, right? Between the cell of the blood vessel, right? Get into inside, and then Cryptococcus magnify themselves, to protect themselves from phagocytosis. Yeah. Then go through another part of the blood vessel. Then go to where? The target organ, to brain and meninges. Okay, this is just found in only several years ago. Actually, Cryptococcus doesn't have a high technology in 10 years, but within these 5 to 10 years, we have a very new information about the Cryptococcus. Here we have the diagnosis. Yes, CSF, because Cryptococcus have the capsule. Candida have a capsule or not? No, only one yeast up to now have the capsule. It's Cryptococcus. So by this special property, so we can apply in the ink preparation as a direct examination to contrast the cell and stain the background. I will show you later. Not only the CSF, serum and plasma also can be checked for the cryptococcal antigen. What is a cryptococcal antigen to be tested by the serology? We test the capsule, okay? And the WHO, the bedside recommended test is lateral flow assay as a screening test, or we call bedside test. It can be only less than 30 minutes. We can say that this sample have Cryptococcus or not. Very high specificity and high sensitivity test. And another one is culture. For the antifungal therapy, use amphotericin B and fluconazole. But unfortunately, Thailand, we do not have the 5-FC, 5-fluorocytosine, which is quite good for treat Cryptococcus. We hope that in the near future, we will have the 5-FC for the treatment. That you will study in the pharmacology, what is the 5-FC action. C. cactii and C. neoformans are a bit different of the natural habitat. Yeah. For the root of infection is the same, but for the risk factors, C. cactii is still unknown. We still have no idea, no definite idea. But for C. neoformans in here means A, D, and AD. It means Grubii and neoformans variety. Normally, HIV, cancer, and corticosteroid. Recently, within these 5 years, as far as I concern, yes, HIV is now less and more non-HIV become increasing as a host for cryptococcosis infection. Disease C. neoformans will be meningoencephalitis, whereas C. cactii mostly is pulmonary. However, it became a foci as a gone in the brain and meninges too. Can you see in this slide? You can see, right? Yeah. Just focus on the red box. Cryptococcus CD4 less than 100 cells. And other disease, you can see here, a bit different. And this is one of the guide. Yeah, when the physician see the patient, some of the recommended guide. And this slide is quite good for you to summarize all the disease of the cryptococcosis, histoplasmosis, and tuberculomycosis at the end of the column, the last column. You have to differentiate histoplasmosis and tuberculomycosis, because histoplasmosis and tuberculomycosis, which we are going to study later on, yes, two of these diseases are very similar. We call brother and sister disease. But cryptococcus is caused by the yeast. Yeah. 3 of these diseases have the skin manifestation quite similar. So we have to differentiate by the direct examination, by the staining, and by the culture. Okay? 3 of these diseases. That's why I put 3 this slide to show you. Escape of the host defense. Yes. For the xenoformans, complement system will be very helpful. Downregulation of the antigen by the macrophage. Immunosuppressed by the cytokine production. And stimulation of the suppressor cells. Its capsule is one of the resist of the protective mechanism of the cryptococcus. Here, I can show you. Capsule as the outside green. This capsule contains polysaccharide. For the polysaccharide, is a good antigenic property or not? No. Right? It's a very inert. This polysaccharide have the galactosyromannan and glucurosyromannan. The high incidence, the high percentage of the GXM contained in the capsule. Okay? The serotype A, D, B, C, A, D differentiated by the structure of the GXM and GAL-XM. Okay. Lateral flow assay, base site serological test for Cryptococcus, detect this capsular antigen ได้นะคะใกล้จะหลับหมดแล้วได้นะ Okay. This test I show in here only less than 15 minutes. It's a strip test, very easy. You just put the specimen and then drop the solution, then put the strip inside, insert inside. Then they have a standard and then test it. What is on the strip to show you that the serum is positive for Cryptococcus? This strip check antigen. So my question is that what is the compound that is on the strip to show you that Cryptococcus is positive? Antibody, right? The antibody will be coated on the strip, to test the cryptococcal antigen. And here, this is in the ink preparation. Stain the background and contrast with the capsule. When we get the specimen, we don't know whether the patient was infected by the fungus or not. Sometimes the specimen sent to the bacterial laboratory. And this is acid-fast staining. You can see something like a halo surrounding. We cannot say this is a Cryptococcus. We just suspect it. It might be Cryptococcus. We have to confirm by the ink preparation or staining the capsule. So please go back and find what is the staining for the capsular antigen in histopathology. I'm not going to tell you, but I can give you an exam. Everyone got it, right? It will stain with the red color, and it will stain the mucilaginous capsular antigen. And this is the slide that shows you the pathogenesis of the Cryptococcus. When inhale, okay? When we inhale the cell, it will go to the lung. And after the lung, it will be spread to the brain and meninges and to the lymphatic root and lymphatic, to the lymph node and to the skin. Here, this is the blood vessel, okay? And this is lung alveoli. After the macrophage, this is macrophage. This one with all the ray surrounding is the yeast cell, it's a Cryptococcus. When the yeast get into the body, then the macrophage will be phagocytosed. Some giant cell is working here. But when phagocytosed, the yeast will be inside the macrophage, like this. Then, when the cell, macrophage is a lot, right? And macrophage, it die or not. It has a long life. But when macrophage explode, ระเบิดออกมาเพราะฉะนั้น macrophage Cryptococcus from this macrophage will move into another macrophage, right? And the macrophage will move surrounding, and sometimes move into here, into the bloodstream, and can show you in these here. It can go into the blood-brain barrier here. the macrophage will carry a lot of cryptococcus and go through the bloodstream. And after go to the bloodstream, it can explode out and infect directly to the tissue at the bloodstream. Because in the blood, in the brain, we have the substrate for the cryptococcus. The substrate on the brain will be the diphenyl compound. The substrate for the melanin production. Cryptococcus have the lactase gene or phenol oxidase enzyme to use the substrate on the brain and meninges as nutrition for growth. Then produce melanin pigment. The melanin is one of the virulence factor, right? So melanin will protect cryptococcus from being killed by the immune system. Okay, I think we can pass this one. This is just some gift if someone want to study. This is the study in Sahara, in Africa, and to show you how important of the prevention or the early detection of the cryptococcus infected human body. Then if they have the early detection, the patient will be treated in early phase, then lifelong comprehensive HIV will be more. So this one just prevention. If have the good prevention, then the treatment doesn't have so long. ถ้ามีการป้องกันที่ดีถูกไหมคะ treatment ก่อน เพราะฉะนั้นการค่าใช้จ่ายต่างๆมันก็จะลดลงนะอันนี้เป็นสิ่งที่สำคัญมากเพราะฉะนั้นเขาก็จะเริ่มไปทำที่ต้องหา population ใหญ่ๆอะค่ะมาทำในบ้านเราไม่สำเร็จเพราะบ้านเรา population คนเกิดโรคมันน้อยนะคะนี่ก็เป็นสิ่งที่เขาก็จะพูดเรื่องของ opportunity benefit and challenge นะ for prevention and how about the treatment at the same time and compare what is useful to prevent and what is the one of the virulence factor of the cryptococcus. What is the virulence factor of the cryptococcus? Capsule. Then another one is melanin pigment. The mechanism of the capsule, it prevents the UV light. Okay? Then it prevents what? It prevents what? You can see here. It prevents what? It produces peroxidase, right? Some free radical. Yeah? It produces some free radical. So free radical will not kill cryptococcus. The microbe is very clever to prevent themselves. Now it's already. This is might be too much in detail, but this on the right hand, this one you can study by yourself. Yeah. The one I would like to mention is virulence factor. They have a capsule, they have a lactase gene, and two of these very useful to apply for the diagnostic test. Like here, phenol oxidase, we can use diphenyl compound as a substrate. Then colony will become dark, so we can differentiate from other yeasts. For the Titan cell production, the Titan cell production means when the cell get invade into, not invade, when the macrophage phagocytose, the cryptococcus, the cell size will become bigger, and many nucleus is inside, and have a large vacuole, and thick cell wall. So all of these properties, all of this structure prevent themselves from phagocytosis, from being killed by the macrophage mechanism. Here you can see KOH preparation with a high capsule, dark DGMS, and the color of the colony is not like a candida. Candida is rather white, but for the Cryptococcus is rather yellowish. Okay, so we finished Candida and Cryptococcus. Now we come into systemic infection. Similar, we divided into monomorphic and dimorphic fungi. For the monomorphic, for example, septate hyphae and non-septate hyphae. Then dimorphic fungi have an endemic zone, as mentioned at the first of the first slide. Here, thermodimorphic fungi. In Thailand, we have two diseases, histoplasmosis and tularomycosis. Tularomycosis, it used to be the name as Penicillosis maneffii, but right now they changed the name of the genus. So the name of the disease will accordingly change to be tularomycosis. Coccidioidomycosis, blastomycosis, and paracoccidioidomycosis. Three of these diseases are found in USA. The tissue form of each disease are different. For the paracoccidioidomycosis, just like ลืมแล้วเขาเรียกว่าอะไรพวงมาลัยเรือค่ะพี่ลืมไปแล้วเรียกภาษาอังกฤษว่าอะไรนะไม่ช่างมันเป็น multiple budding surrounding the big spherical like this. For blastomycosis, it has a wide septate between mother cell and daughter cell. We call broad-based bud. The size is 2 to 10 micron, like this. I found one case in the cat. Even we don't have any human case right now, no reported case. We are very alert because we never have blastomycosis in Thailand. However, I found one case with Kasetsart University's veterinary. For the coccidioidal, this one just a model to show you that for the systemic infection, especially the thermodimorphic, they will get the inhalation. This is the environmental area. The fungus have a spore, very small spore here, then spread into the air, inhale into the pulmonary, and then get infected in the body. This is the model of the systemic infection in terms of the thermodimorphic fungi. The first disease will be histoplasmosis. This picture include and conclude everything. If you remember this one, in this cave, in this frame, in the environmental, the source will be the soil, contaminated soil with the avian or high nitrogen source. And in the natural habitat, the bat, ค้างคาวค่ะ will be another source. And the shape of the fungus will be the mole, have a big spore and spiny spore. Okay, when the spore, the small spore, we inhale the small spore inside the pulmonary here into the lung. This fungus, this mole, histoplasma, will be phagocytosed in the macrophage or in the giant cell. We call them intracellular infected cell.